You will now be placed into the conference.
Obviously, with the worldwide pandemic, we've had to
Muted.
our annual conference for the International Conference for Diseases in Nature that are communicable to humans, and which would normally be held this year in June in Toronto. And we had so many amazing abstracts come in that we thought that it was something that we'd like to share with everybody, regardless of whether or not we could be there in person. So we're really excited about the number of people who have decided to join us today, and we hope that you'll continue to join us on a weekly basis. If there's something that you feel that you would also like to present, we are willing to continue to add seminars right on through the entire summer. So far we have sessions set up until about the end of July. Just as a housekeeping measure, we do have all of you on mute. There's just going to be more than 100 people on. And so the way we are going to handle it today is that Jay is going to be doing his presentation. You can send me questions, and once the question and answer session starts, then I am going to ask him the questions in order to stimulate further discussion. Jay thought that it would be best to have a very short presentation on where research currently is, and then welcome all of your questions, just as far as what background information you want to know, or any of the research that's going on, that kind of thing. So just to introduce Arinjay, it is my pleasure to introduce an amazing researcher and a very dear friend of mine, Arinjay Banerjee. He is currently a postdoc at McMaster at the Institute for Infectious Disease, and has been working with Dr. Samira Mubaraka with Sunnybrook very much since the beginning of March. Ironically, the three of us were all on the planning committee for INCI, and so the whole initial research project started as part of just a very generic conversation that we were having during the beginning of the outbreak. So I'm going to let him start because he has way more important things to say than I do, and thank you all for coming. Sure, over to you, Jay.
Awesome. Thanks, Jasmine. I think we're a little tight on time, so I'll get right into it, and hopefully we'll have 15 minutes for questions. It's a very small presentation, and I'm just going to show you guys how we came together, what we did, and the science that we're doing. So before I dive into my slides, what you're looking at is an electron micrograph of human lung cells infected with SARS coronavirus 2. And the little vesicle that you see, a little round circle with lots of tiny black dots, those are SARS coronavirus particles inside the cell. Okay, our next slide, please, Andrea. So what did we do when, like Jasmine mentioned, we were on a meeting phone call, and Samira mentioned that Canada's got one of its first COVID-19 cases. This was early March or late February, and I want to say late February. So we instantly decided we should go back and collect a specimen from this patient and get the virus out. Because having the virus gives us the ability to develop diagnostics, vaccines, and therapeutic drugs. So we went in, we collected samples from two COVID-19 patients, and we immediately took it to a high containment lab at the University of Toronto, and we put them on immunocompromised cells. Now, Vero cells are African green monkey kidney cells, and they lack a functional immune response. That gives us an opportunity to isolate viruses from patients. And once we had the virus isolated, we did the diagnostics on it, the virus sequence using two different sequencing platforms. Next slide, please. So this is what your cells would look like once it's infected with the SARS.
Jay, just a moment. Some people are asking that you just speak a little bit louder, so if you can just...
Okay. All right. Does that help? Assume that's working. If that's a problem, I'll keep an eye on the chat. Okay. So what you see on the left is mock inoculated samples. So we always need a control that tells us if cells are dying because they're contaminated. And we had clinical specimens from 2 COVID-19 cases in Canada. And we took the specimens and we put them on these immunocompromised cells. What you can see, and I hope you can appreciate, is the enormous amount of cell death in the middle panel and the right panel. So we instantly knew that we had isolated something. Next slide, please. So we then did a bunch of confirmatory tests. We sequenced the virus. We know the whole genome of the virus. We know the virus that's circulating in Canada. We also took these cells and we imaged them using electron microscopy. So what that lets you do is that lets you super magnify what's inside the cells and lets you visualize the viruses. So this image is 36,000 times magnified. And this was my opening slide. So the red view, there's a vesicle inside these cells that's full of coronaviruses. Next slide, please, Andrea. There you go. And if you zoom into these virus particles, you can. It's not very distinct, but you can look at the glycoproteins, what we call the spike proteins. And because these viruses have a spike protein coater on them, we call them. I'm still seeing this. Okay, we've probably fixed the volume issue. So if you look at the spike protein, that's why you call them a coronavirus, because it seems like it's got a crown around the virus protein. Next slide, please. The next question we wanted to ask was, what cell types do these viruses infect? It's absolutely critical to identify the cell types that a coronavirus or any new virus can infect. Because if the virus is able to infect immune cells, that's very bad news from a vaccine perspective. So we tested a bunch of cell lines from humans as well as monkey cells. And I won't get into the details of this, but what you see on the y-axis is the amount of virus that's propagating in these cells. What you see on the x-axis is a whole bunch of different cell types. And so Calu-3 cells are human lung cell lines, and the virus does replicate very efficiently in these cells. Now, if you look at PBMCs and THP1 cells, these are all immune cell populations in humans. And thankfully, none of these cell types have a productive infection. We do see the virus entering CD4 T cells. So one of the adaptive immune cell populations called T cells, the virus does enter these cells. The virus does not come out of these cells. So that's good news. We don't know what virus entry is doing to the cells, and these are questions we are testing as we speak. Next slide, please. So the other studies that we have ongoing, and I wanted to share this with this community, that we are actively working on this project, and we're trying to answer a lot of questions that will help in developing drugs and vaccine efficacy as well. So we've taken patient samples, we've isolated it, and we now have 2 different isolates for the SARS coronavirus. We've also sequenced these isolates, we've confirmed it, and we've shared it across Canada to facilitate vaccine trials and drug testing across Canadian universities. What we are doing at the moment, we're also testing drugs in-house in vitro. So we've taken cells that can be infected. So we're testing drug targets on these cells that we know can be infected. And we are working on the protocols to get animal testing up and running as quickly as we can. Things are a little difficult working in high containment labs. Everything that you touch and move in a high containment lab has to be approved for safety. Next slide, please. I won't get into the details of this, but what we have been able to do is generate an immune response profile in infected human cells. So we have experimentally infected human cell lines in the lab, and we've identified immune responses that are activated and immune responses that are suppressed. So we need to be able to identify what the virus is tweaking in the human cells that makes it so easily transmissible. Is human immune response efficient to control replication of this virus? So these are questions that we are testing. Next slide, please. Yeah, okay, so I just wanted to keep it super brief so we could spend more time taking questions. So this This is the team on the left is a team at U of T in Toronto that's actively working on the project. We've got some brilliant techs and PhD students who are helping out. And the picture on the right is Samira, Rob, and myself, who really came together. Like Rob used this great example of coming together at the grassroots level. And that's what we did. We all came together, got the patient samples, got the virus out, and we've now shared it to facilitate science across Canada. Next slide, please. Obviously, this was a huge collaboration between McMaster University, U of T, and Sunnybrook, and none of this could have been done without funding for reagent supplies and CL3 labs, which are very expensive to maintain. So CIHR and NSERC have funded us. We are now getting a lot of funding from other groups as well. It's never enough, but it's obviously very helpful. I think I'll just take questions.
Thank you, Jay. That was great. It gave us a little bit of some background there. What are your next steps? What is the team envisioning as far as sort of 3 months from now, 6 months from now, a year from now?
So some of the stuff we're doing is testing drug candidates. Obviously, we've identified cells that are susceptible. That was the first step, really. If you have to test your drug, you need to be able to establish a good model to test your drugs on. So we've done those preliminary studies. We are diving into testing some drugs. We also need to understand the molecular mechanisms that are modulated by the virus. You cannot fight what you don't understand. So we are trying to still really understand this pathogen, understand how it interacts with human cells, and understand how it interacts with the immune response in human cells.
Okay. And so we know that feline and ferret models have been used quite extensively for SARS and for other coronaviruses. Is that where the animal models have been focused, or have there been other models used as well to date?
So we know that Vito Interpack's been doing some great science with the ferret model. So I haven't seen the data yet, but from what I've heard on news outlets, that they have established the ferret models. But there are groups working with Syrian hamster models and mouse models. Syrian hamster models were an excellent model for SARS-1. So it'll be interesting to see what comes out of the hamster models as well.
Okay. And as an expert on bats, and I know that we've had numerous conversations and there's been all kinds of banding around about the role bats have played. What is the research so far, and what is your opinion on how closely bats are responsible for all of this?
So the evidence that this virus jumped directly from bats to humans does not exist, right? So it's anecdotal. We know the virus evolved in bats. There's likely an intermediate species that we haven't caught yet. And the pangolin story is interesting, but it does not fully identify themselves as, it does not identify pangolins as intermediate species. So we're definitely missing something, right? So having said that, we are working on bat cells. We are trying to infect bat cells just to understand if this virus is capable of infecting and propagating the bat cells that exist.
And so what are you considering to be the most fascinating part of all of this research?
I think what's fascinating and also overwhelming is the amount of information that's coming out every day. There's so much we're learning at the time globally, but so much is being put out on preprint servers in real time that it's almost impossible to just keep up with the literature, I think. So I think I'm just kind of focusing on we know of groups that have been working on coronaviruses for extensive periods of time. So you know people who are doing fundamental science and people who are doing other kinds of science. So I guess there's some vetting process involved in the community where you know which science to catch up on.
So then we know that depending on which country you look at, there have been differing numbers of strains of the virus. So in some countries, there seems to have been sort of three major ones. Iceland has come up, I don't know, it's more than 43. So how fast does this virus seem to be mutating, and do you think it is becoming more virulent, or do you think that it's actually starting to ease up a little bit?
So, that's a great point, Jasmine, but the reality is we just don't know. Coronaviruses exist as quasi-species. They exist as multiple populations. Even in a tube in a laboratory, when you sequence the stock virus, you always find 2 or 3 populations. That's just the nature of how the virus replicates in cells. And that's why I think, or some of us think, it's critical to keep isolating this different isolates. The more isolates you have, the more you can do pathogenicity studies. You can see if your isolates are evolving. You can also test serum antibodies from patients who have recovered and see if they continue to neutralize the new viruses. And honestly, we don't even know if these are new viruses. We are assuming that these tiny little mutations are sufficient enough to call them a new virus. Now, we are all learning about these things in real time.
Okay. And are there specific animal species that you think are going to be more susceptible to the virus that we don't yet know about?
So this was recently looked at. It's not an extensive list of animal species the researchers looked at, but it's better than nothing. So of all the animals we looked at, cats are susceptible to SARS coronavirus 2. The study showed that cats can be infected and cats can transmit to naive cats. The study also showed that dogs, ducks, chickens, and pigs were not infected. And they obviously also identified that ferrets can be infected. So you can assume that animals that are closely associated with ferrets may also be infected, although this is just speculation. And one group in Germany looked at bats, one species of bats. Now, bats are over 1,400 different species, and we know that all of them are immunologically very distinct and different. Right? So bats are transiently infected. So we don't fully understand if bats can spread the virus to other species of bats, because bats are always co-roosting together across different species. So, and none of the wildlife's been tested, and that's something I've been talking to some veterinarians, and maybe this community can really push that question that what wildlife species in Canada can get infected. I think it is critical to understand if the virus can go from humans to animals, and if the virus can persist in animal populations, because that's a risk that could exist, but if you don't do the studies, you never find out.
So then you mentioned the vaccine that's being worked on out of Vito. Do you have any information on any of the other vaccines, for example, the one out of Oxford or the one out of Berlin that are being tested and produced?
Yeah, so there's a bunch of different vaccines that are being tested. So you mentioned Oxford. There's also right? But then the good thing is the scientific community is racing towards developing a vaccine. Not just a few groups, but more than dozens of groups are trying to develop a vaccine. And the reality is, as you go along the vaccine development pipeline, a lot of these vaccines are going to drop off, right? Like maybe 95% of them will drop off. But we're hoping that at least a few candidates will make it past the vaccine testing pipeline and will eventually have a few good options to pick from.
And so there's been all kinds of debate with regards to the immunity that individuals develop and whether or not there appear to be some individuals who do not maintain their immunity after having been infected. Do we know if that is common or if it just has to do with like a few specific individuals or maybe poor science or conspiracy theories, that kind of thing. And so if they do develop immunity, exactly how protective that is?
So the studies that I've looked at antibody responses in recovered patients have identified antibodies that cross-react to different antigens in the virus. In fact, a very recent discussion with a professor at Yale, they've identified antigens, different proteins in the coronavirus that are more immunogenic. So which proteins in the virus induces a more robust antiviral response? So these are all very effective for diagnosis. You can look at antibodies in patient serum and you can identify if they've seen the virus. But you ask a very important question. Having an antibody response does not guarantee that you're protected. You have to do studies on how effective is your antibody in neutralizing the coronavirus. Now, you've also raised a point on the coronavirus acquiring mutations. Now, unless we isolate these viruses and we continue to test them against serum from recovered patients, we do not know if the antibodies from the first set of exposures are still protective against any strain that might adapt in humans. These are all outstanding questions, and I think as we progress through the pandemic, we'll have some answers to these.
Okay, so there is the concern, obviously, that if the virus is mutating as fast as it appears to be in the media, that by the time we do get a vaccine created and tested, that it will no longer actually provide immunity to those individuals who are still susceptible. How likely do you think that is?
I think one thing that I want to make very clear is the virus is not mutating very rapidly. That's just not true. The virus so far has been fairly stable across all the isolates and the database. What is happening is we're identifying this very tiny little mutations. Now, we don't know the functional outcome of those mutations. Like I mentioned, coronaviruses do exist as multiple populations, even in an infected petri dish of split cells. So it's not that the virus is rapidly evolving or it's rapidly mutating. It's possible that it's like in theory, every virus will adapt. The more time it spends in the human population, it's going to adapt. But we don't know if these mutations are making it more pathogenic or if it's attenuating its pathogenicity. It could go either ways, right? So I think the more isolates we have, the more studies we do, we'll figure these out.
Okay, and just a question, obviously, with regards to the potential danger of it getting into our own wildlife. Which species of bat have been tested, and do we actually have those species in the wild here? And what do you think the potential is for either our mustelid population or our felid population or our bat population becoming infected and it becoming an ongoing problem here in Canada?
So the only species that's been tested, I think, I'm speculating on this. I've forgotten the species of the bat, but I think it was a fruit bat species, and I could be wrong. I think the Germans tested it, and they saw that fruit bats can be transiently infected. Now, we know that majority of the bats in Canada, hibernating bat species, are insectivorous bats. These are very distinct, very diverse from fruit bats, right? So can fruit bats be infected? Can insectivorous bats be infected? I know at least at a cell line level, we've tried infecting insectivorous bat cells. And these are kidney cells in a petri dish. They cannot be infected. But that does not mean that bats as an organism cannot be infected. That still remains to be tested. And as far as wild animals are concerned, you'd likely want to survey and monitor animals from the cat family, because we now know this is widely known, a tiger was infected in Bronx. And we also know that domestic cats can be infected. So it does seem like cats do express a receptor that might make them susceptible to SARS-2 infections. But we don't understand, like nobody's tested the full breadth of wildlife in Canada or anywhere else that can get infected. So these are questions that have to be tested.
Okay, and so then with respect to that, given the fact that we have a number of species at risk or endangered animal species, how important do you feel that it is that we find a multi-species type of vaccine?
So, like, developing a vaccine is one question, right? So let's assume best case scenario, we have a vaccine that's immunogenic, that's protective, and safe in multiple species. Now think about the complications associated with implementing that vaccine. How do you vaccinate wildlife species? How do you vaccinate bats? Like that's going to be it's crazy. I can't even imagine developing a strategy to vaccinate wildlife with a coronavirus vaccine, right? So we might be better off just focusing on humans because we seem to be more easily infected and we easily transmit this virus. So if we have a vaccine for humans, we're less likely to transmit this to other animals.
Okay, and so then as you know, there are multiple coronaviruses, and most species have at least one that percolates through their population at any given time. With respect to pigs, we've had PED in the population for an extended period of time, and there has not been a successful vaccine developed. So do you think that that is more part of the money being poured into it? Obviously, with it being humans, there's a lot of money being poured into a vaccine for this. Or do you think it has to do with structure of vaccine? Give us your thoughts.
For post-epidemic diarrhea virus, or PEDV, Vito Enterprise did make a vaccine. And there were other vaccine candidates. I'm not 100% sure how effective these vaccines are in pigs. But vaccines do exist. Coronavirus vaccines for PED do exist. But you're right. I think there hasn't been as much effort for animal coronavirus vaccines as we are now starting to see for human SARS coronavirus too.
And then with respect to just symptomology, there appears to be worsening in some populations, for example, those in New York that are becoming infected. Do you think that this is actually true worsening of the symptoms, or is there bias in reporting, or do you think it's part of the reality of there being reduced supportive care because there are so many cases? Can you speak to that at all?
So I don't think there's a lot of regional correlation in Canada in terms of number of cases. Obviously, some provinces are seeing less. Depends on the population demographics. It depends on how densely packed a province is. So you'll notice that Ontario and Quebec have the highest number of cases, right? Obviously, in Ontario, we have a lot more people living in close proximity, sharing transit services, sharing a lot of things. But what does seem to predispose, and this is based on literature, what does seem to predispose people to a more bad outcome with COVID-19 is the elderly population seems to be more susceptible, right? If you have underlying health conditions, if you're immunocompromised. So these are populations that they should be more careful going out, because we know that data is out there that tells you that these individuals are more susceptible for a bad disease outcome.
Can you speak at all to the new symptom of sudden stroke that appears to have occurred in middle-aged people?
Yeah, so we are working with Jay Hirota at MAC, who's trying to identify what cell types viruses have infected in COVID-19 patients. And not just cell types. Do you find the virus in heart? Do you find the virus in lungs? Now, again, I haven't seen a peer-reviewed paper on the stroke. I did read the news piece that talked about increased stroke in younger populations. But unless I look at a peer-reviewed paper that's been reviewed and accepted by experts in that field, I think it's very difficult to comment. There's just so much coming out.
Okay, and then this will end up being the last question, because I know you have to go. You have another another session almost immediately. But what about the potential infection of other species such as birds or reptiles or amphibians? Do we need to be concerned about anything that's non-mammalian?
So we don't have any data that human coronaviruses can infect non-mammals. But there are other coronaviruses that can infect these reptiles, for example, or other species. But I think as far as SARS coronavirus 2 is concerned, we should restrict ourselves to mammals that are closely associated with humans. We don't know if, especially monkey populations as well, which fortunately we don't have out in the wild in Canada. But from the Canadian perspective, I think especially monitoring cats. And I've been trying to stress on domestic cats, keep them inside, don't let them walk out, because you don't know who the cats are or what the cats are interacting with.
Right.
And there's data out there that will tell you that cats are susceptible to this infection.
Okay. So, Jay, unfortunately, I know you have to go. So what we'll do is if anybody has any further questions, feel free to send them either to Andrea or myself, and we will send them on to Jay and have him answer them, and we can post those later on our website. and potentially send them back to you if you've actually emailed them to us. But if everybody could join me in just thanking Arinjay for taking the time to speak to us today. And Arinjay, good luck and stay safe, okay?
All right, thanks, Jasmine. Thanks, Andrea.
Bye, everyone.